All right. Well, hi everybody. Thanks for attending. This is lesson 5 of functional annotation, and it's sort of part of a set of lessons that followed Ben's lesson 3, in which he did a good job of laying the foundation for using HMMs to annotate data sets. So I thought it'd be appropriate to have a couple of slides just to briefly mention the different databases that are available that house HMMs. Actually, before I begin, I should probably make a quick note that there's 2 other instructors on this channel, Dan Speth and Adelaide Rhodes. So thank you guys for attending. Also, quick note, if anybody has questions during my talk, feel free to ask those questions in the chat. I'm happy to answer. Okay, so yeah, so there's a couple of databases that I wanted to sort of shine the spotlight on. There's quite a few of them. Hummer actually has a really nice web server interface where you can do a number of things if you have a sequence or if you have an HMM. And you can paste the sequence or you can upload a file, and this file can be either a protein sequence, as in the case of HMM scam, or it could be a hidden Markov model or an HMM. And if you click on the HMM search, you can use this profile to search against sequence databases. Like the next slide will show you, if you hit HMM search, you want to provide an HMM, or you can upload an alignment, and you can then select a database that you can search that model against. And that could be a reference proteome, UniPro KB, SwissPro. So all of these really great repositories. I'll shine the spotlight on PFAM and TIGRFAM just because those are the 2 that I use most commonly. Other ones are Gene3D, Superfamily, ParaSF, and TreeFam. I haven't used those, so I can't say too much about them. I'm sure you can look up their information on the internet, or if anybody else here, any of the other instructors has anything else to say about those families, feel free to chime in. But yeah, so you have these at your you have these available on the Hummer web server, and the web server is really great. And once I probably should have included an output, what an output looks like, and it's really great. You can do a lot of things, and you can download all the sequences that were identified. You can download the HMMs. You can look at the phylogenetic distribution of all the hits that you get to a particular protein or an HMM. So it's a lot of really useful, great things here that I really like. Okay, so I mentioned this briefly, and I apologize for those that attended the Genie lecture that are going to have to see these crappy slides again. But I kind of wanted to briefly jump back in and just remind everybody of how easy it is to get an HMM from PFAM. And one of the HMMs that's there is the 7-transmembrane receptor, the rhodopsin family. So if you can fairly easily find this, if you simply type rhodopsin in the keyword search, and on this is going to be the first result that comes up, and you click on that, and you get this really nice page. You can see the tertiary structure of the domain here. If you click on curation model, it takes you to this page, and then you have the availability to download the raw HMM for this family. And that HMM is going to include the gathering cutoff, the trusted cutoff, and the noise cutoffs for that. Yeah. So TIGRFAMs is another great repository that I frequently use, and we have some really high quality HMMs on this in this repository. So if you click on any one of these, and there's thousands of these, this is just showing the first few dozen or so. So if you click on the first HMM, for example, for the ribosomal protein BL35, you can see that there's a number of there's a bunch of information about this HMM, including the citation. So you can actually copy and paste this link into your Google search and read this paper and find out more about this family, about this gene. One thing I will note is that PFAM is actually domain-based HMMs, while TIGRFAMs is, as far as I know, it's gene-based. Yeah, so you get a lot of information here. You also get a trusted cutoff here. So if you're using the HMM and you're querying it against your data sets, you want to make sure that you look at bit scores above this trusted cutoff. So if you want to download this HMM, you go to seed alignment and you can download the alignment. So downloading an alignment from TIGRFAMs is as good as getting the HMM. This HMM is, has been discussed in lesson 3, and you should refer back to that lesson because if you want more information. But just briefly, an HMM is a probabilistic model of a multiple sequence alignment. Here I'm showing this a very, very simple example, sort of illustrating just 2 positions in an alignment, in a hypothetical alignment. And it's also showing the frequency, the occurrence frequency of various nucleotides in those 2 positions. So then this model can then be used to search sequences and identify probable matches to that sequence. Right. So once you download an alignment, all you have to do is run HMM build, and I'll talk about this. And then once you run that, you'll have your HMM. Right. So this is sort of now getting into something that I like to call the magic lamp part of this presentation. And magic lamp isn't really a published tool yet. It's sort of a pre-existing scaffold, and that scaffold came from Genie. And Genie, as I discussed in lesson 4, is essentially an HMM library of all iron-related genes. So I took over this scaffold and I applied it to a bunch of other HMM collections that I sort of put together. And it's sort of an amalgamation of HMMs that I designed myself and HMMs that I got from TIGRFAMs and PFAM and a couple of other sources. And this is sort of illustrating what's going on within the Magic Lamp sort of broader toolkit. When you run Magic Lamp, you sort of have the option of running Genie. And Genie also exists as a standalone tool, but you also have an access to Wisp Genie, ROS Genie, Magnetar Genie, Gas Genie, and each of these sort of genies have their own collection of HMMs that they use to profile data sets. And the profiling data sets occurs exactly the same in Genie. You need to provide assemblies, and this could be metagenomes or metatranscriptome assemblies or a genome. So the workflow is essentially the same, with the only difference being that you, instead of having one iron gene database or a database of HMMs representing iron genes, you have collections of HMMs representing various other processes. So which pathways are currently supported? That includes biofilm formation, you have reactive oxygen species neutralization, magnetisome formation, a bunch of other ones. I'm constantly adding new ones, just various pathways that I find that I'm interested in. I do my best to find HMMs online if I can. I try to find genes that correspond to a certain pathway and find related homologs that are known to do the same thing, and then make HMMs. Biofilm formation. So all of these HMMs already exist in PFAM. So it was basically just downloading these HMMs and organizing them in a way where Wisp Genie could figure out operon structure, and if it identifies all, for example, all 7 of these HMMs in an operon, including a particular protein that has a certain domain that's really important in biofilm formation, then identifies it as a biofilm formation sort of gene locus or gene neighborhood. And if it identifies these without this particular second messenger, then that, it reports it as something else, right? So It's reactive oxygen species neutralization. Magnetosome formation, I've been, for a long time, I've been fascinated by magnetosome formation and magnetosomes in general. I think it's really cool. So I made a Genie, and this is actually somewhat redundant with Genie because Genie also has all these MAM genes that are involved in formation of these organelles that house basically magnetite crystals. This is the newest one, circadian rhythm, Genie. The name is pretty provisionary at this point, but yeah, this is actually a really cool pathway as well. Because I've always thought that circadian rhythm was just found in eukaryotes, but turns out that there's a bunch of phototropes, cyanobacteria, that are able to regulate gene expression based on nighttime and daytime, and that's sort of related to the redox conditions of the cell, which is in turn a proxy of whether it's making oxygen, whether it's carrying out photosynthesis, or whether it's not doing that. If it's night, then it's fixing nitrogen, right? So this sort of regulation is pretty cool. So I found all the genes that are involved, and it's sort of a multi-locus process that I'm actually still working on. Lethargini is actually built from a bunch of HMMs that are made available by Karthik Anantharaman, who is now a professor at University of Wisconsin at Madison. So these HMMs are publicly available on his GitHub, and I think he actually has a tool that he put out recently that uses these HMMs in addition to a couple of other ones. So all of the ones here that are used by Lethargini, and these focus on respiratory reactions. So respiratory sulfur oxidation and reduction, ion redox, and this is redundant with Genie. And then all the nitrogen pathways, denitrification, anammox, nitrogen, anything nitrogen cycling related. So these are really great HMMs, and Karthik compiled them. A bunch of them were made by Karthik and his collaborators, and other ones were also taken from TIGRFAMs and PFAM. Yeah, so iron cycling, that's, like I said, that's redundant with Genie. Actually, that actually shouldn't be up here because all the iron cycling is my own, except for two HMMs that I got from TIGRFAMs. And manganese oxidation, this is one of the recent ones that I'm still working on figuring out what this means. So from all of these genies, you basically get an output that looks like this. And this is showing Lethargini output first. And basically, it provides a very sort of a broad category overview when you run Lethargini, and you get a bunch of genes for iron, nitriles, arsenic, urea, hydrogen, sulfur. So it'll basically tell you in normalized gene abundance, which represents the size of the dot, how many genes are present in each. In this case, it's a metagenome assembly. And this is data from actually one of Ben Tully's papers from 2017, where he sampled a bunch of fluid from the North Vaughan Aquifer. That's at the Mid-Atlantic Ridge. So that's a really cool metagenome. So yeah, so when you run Lethargini, actually, you can provide a BAM file. So like I said, this is normalized gene abundance. So if there's 5 genes related to sulfur cycling identified in this data set, actually it wouldn't be 5, since this is a metagenome, probably be 500. That would be divided by the total number of ORFs, or gene calls, predicted from this metagenome. And that's what this normalized gene abundance represents. If you provide a BAM file, that will allow Lethargini to take in coverage information, read coverage, and make the size of the dots consistent with that. This is why you see a difference here, because once you make the, you might have 500 genes related to sulfur cycling in this metagenome assembly, but they might be coming from organisms that are part of the rare biosphere, for example, or just not really not abundant in the data set. So the size of the dot would be smaller then, because there wouldn't be a lot of reads mapped into them. Yeah. Another example, and I'm if you can actually expand on these categories, and if you want to see which genes for nitrogen cycling there are, you can rerun Lethargini with another flag, and I'll talk about that flag during the tutorial, and then it'll expand on the nitrogen, and it'll tell you exactly what's there and which assembly. And again, this is normalized gene abundance, not coverage, so this is telling you how many copies of a certain gene are found, normalized to the total number of genes in the data set. And yeah, you can see there's a bunch of NIR-B, NIR-D, there's some NAP-AB, so there's nitrate oxidation going on, nitric oxide reduction going on, nitrate reduction. Yeah, you can rerun it. You get a slightly different dot plot with a BAM file, right? So the distribution of the size, the dot sizes changes when you incorporate coverage information. We do the same thing with sulfur. You see there's a bunch of DSR. There's a SOX, disulfate oxidation pathway. Anyway, you can rerun that with the coverage. Yeah, so HMM Genie, sort of making your own Genie part of the slideshow. HMM Genie is kind of the same, except that instead of having a predetermined collection of HMMs that are included, you can provide your own HMM collection. And I'll talk about how to go about doing that in the tutorial. So the pipeline is exactly the same. It will take this collection of HMMs. You do have to put a little bit of work into putting together this bit score cutoffs file, which is optional. If you don't provide a bit score cutoffs file, then you have an option to set me value cutoff for all your HMMs, and it'll be used for all the HMMs. That's not recommended though, because HMMs differ in their sensitivity. So it's highly advised that you actually do your best to calibrate an HMM and figure out what the optimal bit score cutoff would be. So then you provide this sort of two-column file where the first column is the name of the HMM and the second column is the bit score cutoff for that HMM, and then HMM Genie will use that bit score cutoff, and then it'll profile your data sets. Okay, so that's it. Yeah, that was short enough. So we're going to move on to the Jupyter Binder tutorials now.